or chemical treatment, we talk about this now. You become an extraction process start to occur. Start to get rate behind this is actually what we call as the higher temperature. That's why it become unfold because of the energy being used. This is the water molecules that involve into the system. Okay, so if not that clear, I will try to share in your Putra Blast about the two video. All right. Now, since I think I mentioned to you that we need to get some of the lecture due to our missing 2 hours, so I'm gonna skip this discussion, okay, and also the detail about this DSC because this is more on the technical part. Same as well, the thermal denaturation because it's very much related to the discussion on the DSC. So I'm going to skip that. Okay, make sure, but you can always read. Okay, but we are not going to focus on that because I want to focus more on the theoretical part to make sure that you understand. Yeah. So one of the uh, sentence that I highlighted previously is molten globule state, and I think from our last lecture I also mentioned that this is another keyword or key points. Key points when we discuss about protein stability or folding and unfolding is this Mg, molten globule state. So what is actually molten globule? Basically, it is actually an intermediate. If you look at these, we're talking about entropy, the randomness, then we have the energy. Of course, this is actually the lowest where the native enzyme. Okay, and then this is the glass state, transition state, and this is where most of the molten globule. Yeah, when we talk about the size, it's actually about 10% bigger compared to the native state. It is a compact globule, but yet expanded over a native radius. This is some of the information. So, what else that you need to know about this molten globule? When we talk about the interest to understand the protein folding intermediates, it always lies in their significance to protein folding pathway. So, in this case, molten globule is one intermediate lying on the kinetics and sometimes thermodynamics. Yeah, it can be both way between native and unfolded state. Okay, we know that this is native and this is the unfolded, so it is very important information that we can understand. Yeah, we're talking about the intermediate. We're talking about intermediate. So the molten globule, or maybe after this I just referred it to Mg, is believed to lack the close packing of amino acid side change that characterize the native state of a protein. Then we also see that the transition form, the transition from a denatured state to a molten globule may be they suggested a two-state process that we're going to discuss later on. Okay, so for example, we can use ANS, 1-anilino-8 naphthalene sulfonate, to bind to the protein, and then will give an enhanced fluorescence, and from there we can study. This is actually on how to study this. Basically what we can say is ANS is actually a fluorescent dye. Yeah, so you can get when you use in the spectroscopy, you can monitor. So this strategy is based upon the assumption that when in molten globule state, the exposed hydrophobic clusters of protein are readily bound by the non-polar anilino naphthalene moiety of ANS. Yeah, this is the anilo naphthalene, so it can bound. Of course, in the presence of the brilliant, it will produce a brilliant fluorescence color. Yeah, so that they can what you call this capture the information. Remember, when we look at the video just now, they mentioned that hydrophobic normally exposed at the exterior, while hydrophilic mainly inside. So that's why they want to bind it at the outside, so they can monitor the changes through the fluorescence. Okay, so this is how you do. Then we want to know, we can also do these Mg by looking at using the circular dichroism. Okay, this is actually quite common because it gives the changes in terms of the beta helix, alpha helix and beta sheets. I'm sorry, tersasul lah. Okay, then we can also use NMR proton exchange so that we can see the differences. Yeah. For example, in this information, we can say that a quenched fluorescent signal with broadened NMR chemical peak. So you can have a look at this. For example, the structure, the spectrum of NMR from native state to the unfolded. There are some changes that we can observe, including the molten globule. Okay, so this of course another NMR that we can have a look, but I missed one slide, I think. Okay, sorry. This is what I wanted to talk about from unfolded to molten globule to the folded. So from here, You can also refer to the next slide later on. First, we look at this U to Mg, and U to Mg can be reversible, and Mg to F also can be reversible. That means in this U to Mg, from U to Mg, it may be a continuous transition with how to say this, with no apparent switch from one form to other. Yeah, the folding of some proteins can be modeled as a three-kinetic process. Then F also can be N. Some you may use N, folded or native. In this case, one of the difficulties that, for example, if we want to design, we can call it as a de novo protein design, is achieving the side change packing needed to create a stable native state rather than an ensemble molten globules. So this is actually quite critical. We're talking about the side change packing. Given a desired backbone conformation, side chain packing can be designed using variation of the dead end. Okay, this is slightly complicated. What we call as the dead end elimination algorithm. So don't worry. However, what I would like to say is that attempts to design protein of novel folds having difficulty using this method due to the absence of what we need as what we call the plausible backbone models. So come back to the very simple way how to arrange the amino acid in certain sequence. That is actually the major challenge. But of course, amino acid have its own hydrophilicity or character, and this is why people start to study if they want to create a new protein or new enzyme. But of course, until right now, we cannot beat the nature. Yeah, but there is already many research group in the world start to produce a totally new enzyme or protein, basically from scratch. When I say from scratch, they just use all the amino acid doing the peptide sequencing. Sorry, they do the peptide synthesis that leads to the longer and longer protein. Okay, and there is another way that we're going to discuss next week. We call it as a synthetic biology. We program basically we design certain in synthetic biology. Not going to confuse you with this part. We just focus on this MG. I always have what you call this kind of excitement when I talk about molten globule and protein design. But I'm sure at this point you may get confused with my excitement and with the theoretical discussion. Okay, so we come back to the molten globule. So basically, we can study this one by CD, I mentioned just now, but there are two, far or near circular dichroism spectroscopy. Okay. Let's see what else after this. Yeah, this is what we can see from the folding process, the unfolded to the molten globule. Okay, the unfolded energy is still quite high, but molten globule quite slightly lower. But this is the critical part for it to reach the transition state. If it's achieved, then it becomes the unfolded. So this is, if you look at like kind of this diagram, meaning that you have many conformations, the entropy is very high, is all unfolded. Then molten globule have a maybe less number of unfolded because it start already to form, whether to form the final folded system. Yeah, so the unfolded state is an ensemble of large number of molecules with different conformation. Then it lead to the molten globule. The size might be about 10% bigger. Yeah, because before it start to become totally full, reduce its size until one folded or one native protein or enzyme is actually the final part. Okay. So if you look at this diagram, it's very similar explanation. I'm not gonna repeat all of the information, but you can see this is the unfolded. Then you start to have this molten globule, the transition state. Then at the end you will get the folded. Okay. What we can, the information from here, not all steps have the same rate constant. But when we talk about the kinetic, now from this time we talk more about the thermodynamics, now we're entering into the kinetic. What we can say is every step have its different rate constant because the rate of reaction is very different. The intermediates accumulate also at different concentration, but what they need is only a low concentration. Of course, in details, they identify the kinetic intermediates, and we can measure the rate constant before it gives you to the final part. What we can say other than that, whether it's a fast or slow folding, it depends on the many reasons. Two examples here: formation of disulfide bond. Because disulfide bond, it will create another, for example, cysteine and cysteine. Yeah. Then if there is isomerization, this is very critical because we know that it involves a chirality inside the system. So in short, we can also look at this. Prior to get into the folded, Mg is the key kinetic intermediate. We can look at here, the unfolded to molten globule. There are so many conformation, and it is a very fast. But when come to the totally folded structure, finally is normally quite slow. Yeah, especially from molten globule to unfolded and folded to molten globule. Okay, so we're not going to focus more on the kinetic of this molten globule. I'm just afraid that it will enter into too biochemistry for all of you. But what I believe, until this discussion, you should be able to relate the importance of a protein folding and unfolding, then the importance of studying it, for example, using a denaturing agents. Yeah. At the same time, you also understand there are many other influence that can denature the protein. And of course, in this part, the key player here is also molten globule, other than the folded. We also talk about the entropy and enthalpy that leads to the Gibbs free energy. Now, we also know that denaturation is often irreversible. So one of my friend, Professor Weiss, he was the first chairman of Global Young Academy. I was the first, I think, 60. Global Young Academy means that all the young academics worldwide, we form under the what we call as during the World Economic Forum, I think in 2009. So we agreed every country must have its own young scientist academy or young scientist network. So in Malaysia, we have a young scientist network under the umbrella of Academy of Sciences Malaysia. And I was the one that established that. I was the first founding president and founding member of Young Scientist Network Malaysia and the founding member of Global Young Academy. So basically what we like to highlight is when you become a scientist, we want to make sure that you have a role, just not about doing research, but you also have to speak your voice to the government by involving the scientists in the policy making. We want to write more information. We want also what we call as the science journalism, yeah, meaning that scientists just not do all this kind of technical writing you publish in journals, but we also need to write in the magazine, in the newspaper, so that the public understand what you are doing. Otherwise, scientists just shock sendiri. You're just living in your own cocoon. Okay, so this is quite interesting. We know that we can make a scrambled egg, yeah, from egg, and we basically either boil it or we fry it. But this Professor Weiss from UC in Irvine, so he the first time reversed the reaction, meaning that he can unboil the egg, ungoreng the egg, yeah. Okay, so we know that in egg, when a solution of protein is boiled, the protein frequently become insoluble. Basically, it is denatured and remains insoluble when the solution is cold. So it's actually no way for you to reverse it. So the denaturation of protein of egg white by heat, for example, you are boiling or fried it. Now, the weak forces between charge groups and the weak forces of mutual attraction of non-polar group are disrupted at elevated temperature. So that's why the tertiary structure is lost. Okay, so now can we reverse it? So in this work, I also hope that I can play this video. If not, you just look at how they, yeah, yeah. So boil and then reboil an egg. So look at a very simple experiment. So just collect the egg white. You don't need the yolk for the study. They boil it. Okay. This is very typical. So they use urea. So remember what urea can do? Yeah, urea basically can break all the bonds inside it. Yeah, it's a long process, of course. 16 hours. Basically, they make it panjang, make it the primary structure. So how to refold the protein? Okay, put some urea. It's actually a vortex. It's a mechanical way of doing this. So what happened is, if you imagine that you tarik string along and then you just lepaskan, then it start to fold back on its own. Egg. Okay, so basically what they did, boil, unboil, and boil it again. Yeah. So I hope that this one also you can hear, because this actually the drive behind the research. They're using a mechanical energy. So this is something very similar to what you saw. Yeah. So he's working on the cancer research. Okay, so that's something about it's a motivation, yeah. So I do hope that you also have some kind of motivation in the future in whatever that you want to do. And talking about research, something that related to your understanding. Now, before we end up, I just like to show how all of this information we use it in our research. Okay, this is the example. What we call is a simulation of enzyme in ionic liquids. I think I did mention to you before that ionic liquid is what you call this like the molten salt, yeah, an ion and cation. So we designed it. So we want to know if we can use enzyme instead of using solvent. We know what solvent can do to the protein or enzyme. So we try to use ionic liquids. Yeah, this is the example. And from here, we know that this we need at least 15% of water, because water is still important so that the protein can still do the breathing, yeah, or the movement. We try to reduce it less than that, then what happen is start to shrink and the enzyme start to denature. Basically there is no activity in the chemical reaction. I'm not gonna stress this much. Okay, this is again we study the requirement of water. We can see around here what the requirement is, at least about 15 percentage water, and the extra will be from the ionic liquids. Why we need the ionic liquids? Because when we deal with the substrate, our substrate, sometimes one is polar and one is non-polar. So if you use one particular solvent, it cannot dissolve another one. So ionic liquid have the strength to dissolve both polarity, either is polar or non-polar. Yeah. Okay. That's why the study behind this. Okay. What I would like to show as well, when we start to look at certain ionic liquids. For example, if we use the chlorine, we can see the chlorine ion start to enter into the deep, the what we call as the because chlorine ion is small, so it can penetrate into the core of the enzyme, basically penetrate the shell water molecules. Remember the preferential binding just now, and also hydration. So it can penetrate, and when it's penetrate, it start to interact with the inside. So or we call it as the protein interior. So the effect is we are losing the activity, or the enzyme destabilized or denatured. Okay. So this is why it also leads us to do more on the solvent engineering. The ionic liquid, I also mentioned about the deep eutectic solvent. Yeah, where in this case, this is more on the simulation. We synthesize and crystallize the structure of ionic liquid, and then we like to see that in ionic liquid, basically, forget to put the file. we can see that how this bonding involved as well in stabilizing or denaturing the enzyme that we use. But we also look at other opportunity. This is just the setup of the molecular structure of the ionic liquids. Okay, but the application of it, yeah, instead of we targeted first for the enzymatic reaction, but we found that it may not able to do so for the reaction. Then we also use it in terms of the storing the DNA, yeah, because DNA, when you work with the genomic, when you extract a DNA is so easy or prone to destabilize. So in order to stabilize, you have to put lots of chemicals, yeah, or buffer and so on. So in our work, we thought that why don't we put the ionic liquid, and what we found experimentally and also in the computational, we found that we can achieve thermal stability of DNA, but again, it depends on the solvent thermodynamic, particularly enthalpy and entropy and enthalpy of the water system. So we found that increasing entropy of water will overcome the enthalpy of the stabilization, leading to a pre-melting of the solvent that facilitate the duplex disruption. So in this case, we found that low water content, weaker hydration, more cations bind to the DNA, and DNA it always have groove, so we can monitor the what you call this the location of the ionic liquids that stabilize our in the DNA. Okay, so hopefully this one also work. So this is the example. what I mentioned before, are else able to retain DNA native structure at even slightly higher temperature. This is actually the water molecule. Yeah. So we put the higher percentage of ionic liquid, basically will not affect the DNA conformation. But of course, what we can do, partial dehydration of DNA by ILs prevent the hydrolytic. So meaning that again, it will conserve the structure of the DNA, and hopefully that it can be used in the future. All right, so I think that's enough. It's quite a lengthy discussion on the theoretical part. I think the next week will be less. It's only 1 hour that is still a bit theoretical, because again you need to understand about the folding, about the theory, yeah. And I think the 2 hours lecture later on is more on the application, so that is a bit easier for you to absorb. All right. Now, I've received some of your SEL, so thank you very much, but I'm not able to respond to each yet because of as a dean, my job is like for one day I have 3 or 4 meetings. I'm not like the other lecturers, they have more time. And as well, what you call this, the request for the recording part, yeah. Same as well, I still do not have time to whether to put it in the YouTube, but it's a control YouTube, or try to find the link to put in the Putra Blast. So I hope that this weekend, I hope that I have more time, then I can put the link so that whoever miss some part, but maybe not all lecture, I will select selected lecture. We'll see. All right. Any question that you want to ask me?
No.
It's always the same person that say no. How about the others?
Doctor, may I ask?
Yeah, please.
Now we are, you and your team, right, is searching for the discover how to unboil the egg, right? So how can it be have the application for the protein discovery?
Okay, I think that is a very good question because that will open up to opportunity. What Professor Gregory Weiss mentioned, okay, that they want to use it for cancer research, meaning that they want to understand in the cancer research the protein or the tumor, how they change into such a virulent or such a very bad thing, basically. Yeah, but of course, to work with the cancer cell and then the protein, it will be very costly because you need to use a lot of chemicals. You need, yeah, it's basically expensive. So that's why they start by doing the cheapest way, just buy an egg and then they start to study. And in terms of explaining to people, it is also very easy and very clear. People look at the yeah, most of us basically eat egg. We understand the process and how to reverse it. To all of us, basically, is impossible. But he has proven that it can be done. So that's why they want to use that application in terms of what you call this cancer research. Another thing that I foresee, you may have heard what you call that protein solution, your protein drink and so on. So maybe this would be a new way because we know that when we process the easiest example is collagen. There is no such collagen drink. The moment that you drink, it will kills everything. Not kills everything. What I mean is actually will be disrupted. Yeah. So maybe there is a way using the same concept in collagen. Then only you can take the actual collagen because at the moment when you dry it, collagen basically is gone. So that is how the marketing is actually cheating you. Yeah. When I talk about protein just now, we can also look further into the protein drugs that people have been talking about because the issue with the protein is it is all unstable. Okay, so that is some of the application, but of course the main application still will be in terms of biomedical research, particularly cancer. Right?
Okay, thank you, doctor.
You're welcome. All right, so hopefully that you start to think about your lunch or maybe brunch now. I'm thinking about my brunch as well. So please enjoy your weekends. Don't forget that you only have few more hours to submit your SEL. And I think I also post some question, yeah, in the Putra Blast. Okay, so see you next week. Have a good day ahead and have an awesome weekend. Bye.